Good morning, my name is Anmulhan and today I'll be presenting on Isoniazid. Let's start with the origin of Isoniazid, commonly known as INH. It was first synthesized in 1912, but it wasn't until the 1950s that it gained widespread use after proving highly effective against tuberculosis or TB. Isoniazid is a synthetic antimicrobial drug, meaning it is chemically made in laboratories and not derived from natural sources. Its main target is Mycobacterium tuberculosis, the bacterium responsible for TB. INH is used to treat both latent and active TB infections, but for active TB, it must be combined with other TB drugs to prevent resistance. It is usually taken by mouth, but can also be administered through intramuscular injection. Structurally, Isoniazid is also known as isonicotinic acid hydrozide. It has a simple chemical structure made of a pyridine ring attached to a hydrozide group, as shown in figure 2. Its molecular formula is C6H7N3O, and its molecular weight is 137.14 grams per mole. Physically, it appears as a white crystalline powder, soluble in water and stable under normal conditions. Pharmacologically, it is bactericidal against actively growing M. tuberculosis and bacteriostatic, meaning growth inhibiting against dormant cells. Now, let's explore how it works. Isoniazid is a prodrug, meaning it becomes active only after entering the bacterial cell. Inside M. tuberculosis, it is activated by the enzyme CatG, a catalase peroxidase. Once activated, isoniazid disrupts the synthesis of mycolic acids, which are essential components of the TB bacterial cell wall. It forms a complex called the INH-NAD adduct, which inhibits an enzyme known as INHA that is essential for building the cell wall. This damage causes the bacterial cell to lyse and die. Isoniazid is effective against both intracellular and extracellular TB bacteria. It is metabolized in the liver through a process called acetylation. Interestingly, patients can be fast or slow acetylators, which affects how quickly the drug is broken down. Regardless, it is mostly excreted through urine and typically does not require dose adjustments in cases of kidney failure. However, resistance to isoniazid is a growing challenge. This mainly happens due to mutations in two genes, CatG and INHA. The CatG gene is responsible for activating the drug. When it mutates, isoniazid can't be activated and becomes ineffective. The INHA gene, on the other hand, is the actual drug target. Mutations here reduce the drug's ability to bind and do its job. A well-known resistant strain, H37RV, shows CatG mutation. When both CatG and INHA are mutated, it results in high-level resistance, with drug concentration needed exceeding normal treatment levels. A study found that about 86% of resistant strains had mutation in these two regions. Such resistance plays a major role in multi-drug resistant TB, which is a serious global health threat. Finally, let's talk about cost. One of isoniazid's biggest advantages is its affordability. In many countries like Malaysia, it is included in the National Essential Medicines list and provided free of charge to TB patients through public healthcare facilities. This supports the WHO's DOT strategy, which aims to control TB worldwide. In the private sector, isoniazid is available as a single drug or part of a combination therapy. Prices vary, but it remains highly affordable. In low-income countries, a 300 mg tablet can cost as little as 10 to 20 cents. In the United States, a month's supply, 30 tablets, costs around $13. Its low cost and high effectiveness make it a cornerstone of first-line TB treatment globally. And here are my references. And to summarize, isoniazid is a powerful, affordable, and widely used drug in the global fight against TB. Understanding its structure, mechanism, resistance challenges, and accessibility is key to appreciating its role in public health. And therefore, I've come to the end of my presentation. Thank you very much.